Hello, students. Continuing our microbiology PYQ series. Today's very important topic is Coxiella burnetii. It is usually asked as a SAQ. Coxiella burnetii is an obligate intracellular bacterium causing Q fever and shows resistance to adverse environmental conditions. What does Q fever means? It means it's a zoonotic infection, which means from animals to humans infection. And this Coxiella burnetii is highly resistant. It lays for few months without getting destroyed because it is spore like. Also, very few of them are required to cause an infection. That is why the severity and the pathogenicity is very high. So what we would write in the definition that Coxiella burnetii is a small obligate intercellular gram-negative like bacteria that causes Q fevers in humans. What are the important characteristics that obligate intercellular organisms? Also, please remember that it does not require arthropod vector for transmission. Direct inhalation is enough. It exists in spore-like form, which is makes it highly resistant because we know spores are structurally made in a way that they can withstand harsh to harsh conditions. Okay, and which because of its structure, it is poorly stained by gram stain. What are the diseases caused? Q fever, zoonotic infection from animals to human. I have told you in detail. And it causes acute febrile illness. Febrile illness, we know that the body temperature suddenly like raising and feverish like conditions. Like whatever the problems the patient might be having, you would mainly be feeling fever with pneumonia or hepatitis. It is a high cause of atypical pneumonia. Mode of transmission is that simple inhalation of contaminated aerosol from animals. Also, contact with infected animal products. Any both ways infection can happen. What are the laboratory diagnosis? How would you identify? Any serological test, generally IFA. IFA means indirect fluorescence antibody test, or any PCR based methods can be used to identify. Because I will give you one trick. You have to remember that any test, any microorganisms which does not stain properly, like we read above, that it does not stain properly. So here you have PCR. PCR is like a shotgun. You can use it in any situation. The only drawback is it is expensive and time consuming. Otherwise, PCR would solve any of your diagnostic problems. Significance that this Coxiella burnetii is an important cause of atypical pneumonia, and it is an occupational hazard for veterinarians and farmers because these two get frequent contact with infected animal products. So, inclusion: Coxiella burnetii is a clinically important intracellular bacterium causing Q fever with unique resistance properties. I think we have done it properly. See you on the next topic. Thank you.